But today we're going to talk about plasmid curing. Plasmid curing. Now, like I said in the earlier videos, do well to check out these videos, and you see that we've talked about plasmids and what they are. We said that plasmids are extra chromosomal DNA, and they are carrying information, they are carrying genetic information that confer some characteristics on the cell. Now, today we're going to talk about plasmid curing. And what is plasmid curing? Plasmid curing is any process that we use to remove plasmids from within a bacterial cell. There are quite a number of ways through which we can do this, but more importantly, we should ask that why do we want to do plasmid curing? Why is it important once in a while to be able to remove the plasmids that are present within a bacterial cell? Now, there are quite a number of reasons why we might want to do plasmid curing. And one of such reasons is that it helps us to be able to study antibiotics resistance. It's important to know that many of the resistance that are carried by bacterial cells are carried by virtue of the fact that these characteristics are on their plasmids. And so, in order to study how these characteristics work or how the bacteria are able to have this antibiotics resistance, many a times plasmid curing is the way to use to do such studies. We also use plasmid curing for virulence studies. It's also important to know that a lot of virulence factors and virulence genes are carried on plasmids. And so to be able to study these virulence genes and how these genes work or how they function on the bacteria, it's important to do plasmid curing. It's also important to know that plasmid curing can be done when we want to study, biotechnology and industrial microbiology. We also, when we want to study these aspects of microbiology, we do plasmid curing because it allows us, to be able to study the bacteria and know the different characteristics that the bacteria carries and which of the characteristics are important for us and which of these characteristics can be used for other purposes. This is important for us to know. And now it's also important for us to know that sometimes we do plasmid curing in order to look at genetic inheritance. Want to know how genes are being transferred from one generation to another generation in a microbial population or a bacterial population. And in order to do this, plasmid curing is important. So, like I said before, plasmid curing is a way to remove plasmids from within the bacterial cell so that we're able to test out the effect or the functions of these plasmids on the bacterial cell. And this is quite important to note. Now, there are quite a number of ways through which we are able to do this plasmid curing. There are quite a number of ways that we can do this plasmid curing. One of the ways through which we can do plasmid curing, you have biological method, chemical method, physical methods. These are all different methods that we use for plasmid curing. Now, in order to do this plasmid curing, we can look at, for example, physical method. Physical method involves the use of either heat or UV light. The use of heat or UV light is one of the ways we can use to do plasmid curing. Is one of the physical methods of doing plasmid curing. We also have biological methods which involves growing bacterial cell under stress, or the use of our CRISPR, which is our gene editing tool. We can use this is a method of doing plasmid curing, which is a biological method. Now, if it's a chemical method, there are also many methods that we can use, which are under chemical methods, such as we can expose the bacterial cell to novobiocin. Novobiocin is a type of antibiotics, and that antibiotics has a way of disrupting or destroying cell membranes, and so it's able to destroy the plasmids. You can also use orange acridine and ethidium bromide. Orange acridine and ethidium bromide, these are intercalating agents. So they have a way of inserting themselves into a DNA, the DNA double helix. So if you use such agents, it can also help you to be able to carry out plasmid curing. You can also use SDS, sodium deodecyl sulfate. Sodium deodecyl sulfate, SDS, can also be used as an agent for plasmid curing. So all of this is important, and at the end of the day, the important thing is that we are able to study different characteristics of the bacteria when we do plasmid curing. Thank you for listening to this, and remember to like our channel, subscribe to our content, and do have a wonderful day. Thank you.